Mark?
Hello?
Hi. You can start, I guess. Yeah?
Okay.
Thank you for doing that at such a late time as well for you.
Sorry, so could you repeat that, please?
Yeah. Thank you for staying up so late to give the seminar.
That's right.
Okay.
Okay. So thanks everyone for coming. If I become not understandable or something like that, just someone put up their hand or yell out, and I'll try to fix the problem. But I wish I could be there in person. I've just my son is 4 weeks old, and so I had to sort of cancel my plans to come, but I would have loved to have been there. And thanks to all the organisers for inviting me to talk. So for those of you who don't know me, my name's Mark, and up until a couple of years ago, I was working with Radic in Oxford, and now I'm at Monash University in Melbourne. And so what I want to talk to you about today is a theoretical framework that's based on Smoluchowski's theory for how to model stochastic processes of high order reactions, so trimolecular reactions and higher. So just here's just a quick overview of what I want to talk about. I'm going to do a small review of Smoluchowski reaction theory. Most of you will have a fairly good understanding of it, but there are some aspects of it which I think are more arbitrary than you may think about, and I just want to bring them to your attention. And I'm motivated with this problem for two reasons. First of all, when I was with Radic in Oxford, we were talking about how could you use a individual molecule model to simulate a Turing pattern. And more recently, I've been looking at some cell signaling processes, and one that's really important to me is Wnt signaling. And there's a particular challenge with Wnt signaling in doing a stochastic particle-based model that I'll go through. And so hopefully that will give me enough motivation to talk about why I want to solve the problem that I want to solve with generalizing Smoluchowski's reaction theory. And then I'll talk about a couple of test problems that just have some interesting results about them. Okay, so most of you will know Smoluchowski reaction kinetics as the way in which we can model a chemical system by looking at the relative distances between reactants. And when they come within a certain distance of each other, we can then allow them to react. And the rate of reaction on a macroscopic scale will be determined by the distance that we allow those reactions to occur. However, the original theory from Smoluchowski looked at solid spheres, and these solid spheres would move around randomly and then be able to stick to each other. So they had nothing to do with molecules at all in the first place. So in a very simple system, he considered a two-sphere system with a boundary essentially at infinity, but it wasn't really infinity, it was just a very arbitrarily large system. And the two spheres would have two diffusion constants, d1 and d2, and whenever the spheres would come into contact with each other, they would be able to stick. So whenever the distance from the center of the spheres was r1 plus r2, which equaled sigma, so you don't have to remember r1 and r2, then you say that they've stuck. And so he analyzed the Ford-Kolmogorov equation, which is essentially just the heat equation. It's fairly simple. And so if you can look at it in radial coordinates here, where this P is the probability density to find the second sphere in the frame of the first. So this R is actually the separation or the distance between spheres 1 and 2. And so these boundary conditions here, essentially this condition at infinity or at very large distances, just meant that and this is also, the initial condition is also that P is equal to a constant. And it just says that no matter if you wait a significant amount of time, the system is so large that the probability at essentially infinity is not going to change. And this this condition here says that if your distance reaches sigma, then you have an absorb of the state, and so you have essentially a reaction event occurring, and the probability to then find these two spheres freely diffusing is gone to zero. So it's a conditional probability distribution. So here's just a quick animation of the solution to this. It's got an analytical solution, but the solution here is in blue. This is time equals t equals 0. The red is the steady state solution. And I say it's steady state, but there is no real steady state because you only have a sink. But it's a pseudo-steady state, just because it's an infinitely large volume. And so very quickly, it sort of, it doesn't quite reach this distribution. Well, it takes a long time to reach out here, but close to the reaction radius, I've set the reaction radius here to be 1, you get essentially the flux at that point very quickly goes to a constant value. So here is the solution. Capital P now is the total probability of finding the second sphere anywhere in this volume. And of course it's decreasing because you have this absorbing state. And this is essentially the Kolmogorov equation integrated over space. And you have this term here which in very short amount of time sort of disappears, and you're left with what we know is the rate at which the spheres will come into contact. But the important thing here is that you have to wait this very short amount of time. This will become important later on. Okay, so that was Smoluchowski's theory, and he even suggested at the end of his paper in 1917 that there might be some applications to reaction theory. But of course, with molecular reactions, we don't have spheres, we have molecules. But what's important from a modeling sense is that at a macroscopic scale, we don't necessarily care about how individual molecules will fit together, their microphysics, if you like. We don't really care. It's all meaningless to us on a macroscopic system scale. All we care about is the macroscopic rate, reaction rate, which the microscopic physics establishes for us. So if we take a biomolecular reaction, A plus B goes to something, then we can look at all these sort of molecular scale properties, and it's been shown time and time again that it doesn't matter what you really look at. If you look at it on a macroscopic scale, you eventually just get the rate of reaction, or the stochastic rate of reaction, is proportional, so this is law of mass action, proportional to the product of the number of molecules you have, essentially, and this proportionality constant is your rate constant. And this is all we need. So in order to do a good simulation of an entire system, all we need to do is to come up with a set of rules so that we have the right value of K. So here are sort of my two conditions for an accurate simulation of a reaction diffusion system. First of all, you need to have reactants close together. Okay, you can't just say if it's because it's a spatial system as well. You can't just say I'm going to take a molecule from one side of space and then react it with another molecule from another side of space. So they have to be somewhat close together in some definition. And then the second rule is all we need to do is to make sure that no matter what our rule is, that we regenerate this reaction rate K from the specific parameters of our rule. So after looking at Smoluchowski's theory and ignoring the spheres and just looking at the distance between 2 points, it sort of becomes very obvious what our rule should be. We'll react whenever the distance between 2 molecules is less than our carefully chosen reaction radius. So if we look in the frame of molecule A, we have this reaction radius, and if the molecule B comes within that radius, then we'll react. And it's sort of intuitive, and it's intuitive because we've already seen the proof. Smoluchowski did the proof, and we've already seen it, so it's become intuitive for us. But there's no reason why, for example, that can't be a cube, okay? The only reason why we don't say, let's have a look at the individual Cartesian coordinates, is because they're solving this system analytically to try to find out what is the length of this side, for example, that gives us a value of K. It's quite a bit more difficult problem analytically, so we just ignore it and we say, well, the simplest one is the best one. Okay, so I got interested in higher order chemical systems by this Turing pattern example, which is actually from a paper by Yang and Radek a couple of years ago, and essentially it's just a standard Turing pattern. So we have this molecules of U are produced and degraded. Molecules of V are produced, and V can turn into U by this tri-molecular reaction, if you like. So here are the deterministic PDEs. It's fairly straightforward to create these from law of mass action, and you can add in diffusion of these molecules just by adding it in one. This is all in 1D, so it's one-dimensional Gaussian. And so here are the boundary conditions, those zero flux boundary conditions, and we're going to solve it on the domain 0 to 1. So here are the parameters used by Yang and Radek, and this sort of is an inspiring problem that I was looking at. So here is the PDE solution. Of course, it depends on the initial condition a little bit, but if you look at an initial condition which is more or less the unstable uniform steady state and add a little bit of noise, this is what comes up most often. Okay? And we can simulate this actually quite nicely with a spatial Gillespie algorithm, and this is what Yang and Radek did. So if we take a compartment size, spatial Gillespie algorithm, we take a compartment size of 0.025, we end up with these peaks which fairly closely match our PDE solution. So this is the distribution of molecules of U, this is the distribution of molecules of V. However, V diffuses a lot faster. So if you look at the diffusion constant for V, it's a lot faster. So you might think that this compartment size is wasted, and think about putting something that's has a little bit of a larger compartment size. But if you do that, and this is what Yang and Radek showed, you actually destroy the fine detail in the distribution for molecules of U. So it just goes to show that with the spatial Gillespie algorithm, you can run into these artifacts that are due to the discretization of your space. So the question we asked ourselves when I was with Radek was, well, can we solve this without using a lattice? So can we solve this using the Smoluchowski approach? And the first thing that most people ask me when I say I'd like to be able to solve a tri-molecular reaction with the Smoluchowski approach is they say that, well, so here's our tri-molecular reaction. They say that, well, actually, true high-order reactions never really occur. There's some few examples where they do, but in biology they more or less don't. And the reason for this is because if you have U, and V coming together to create 3 U molecules, typically it goes one of 2 ways. Either U joins with U fairly slowly and very quickly recruit V, or U and V react slowly and quickly recruit another U. So you have this essentially cascade of 2 bimolecular reactions. I want to sort of talk about this and why the Smoluchowski approach doesn't really work with the example of wind signaling, which is something that I'm very interested in. So here is a bimolecular diagram of the different reactions that can occur in the wind signaling pathway. It's fairly complicated, so this is just a little cartoon here just to make it a little bit more simple. Essentially, this region in here, the pink region is the cytosol of the cell, and here's the nucleus, and this is the extracellular space. And without Wnt, so here's Wnt, without Wnt, what happens inside the cytosol is that axon, GSK, CK1, but that's usually omitted in models, and APC, they come together and they bind with this molecule beta-catenin, which is very important in the cell. It is responsible for proliferation, differentiation, migration, cell polarities, a lot of things beta-catenin is responsible for. And this complex of axon, GSK, and APC degrade beta-catenin. Essentially, they degrade beta-catenin. This is the degradation cycle here. So you have APC, axon, and GSK binding with beta-catenin, they phosphorylate beta-catenin here, and then beta-catenin dissociates, and this is degraded by proteolysis. And so that's what happens without Wnt. With Wnt, what happens is, actually it's a little bit debated, and I'm looking into how we can resolve this mathematically. But Wnt essentially somehow deactivates this complex. In this particular model, which is the most famous one by Lee et al., they say that what Wnt does is it activates this other molecule in the cytosol, which is disheveled here, and it actually dissociates one of the three components of this, just what's called the destruction complex, from the APC axon GSK, essentially deactivating it so it can't interact with beta-catenin. Beta-catenin builds up within the system and then is allowed to affect the cell in very significant ways. For example, if you take APC out of this system, that's approximately 90% of all colon cancers have an APC mutation. Essentially, the reason why it's so bad is because beta-catenin builds up and tells the cell to do uncontrollable cell division. So this is a really important signaling pathway. But what's important here is that they've done some, this is a paper by Chin-Wei Tan, who's also in Melbourne actually. We're doing some work together. And he looked at the steady state concentrations of these sort of complexes, all these complexes in this system, and he found that, so this is the xenopus egg, which was found from this original paper, but he also looked at HEK cells, which are human kidney cells, and even canine kidney cells, and you can, and others actually, and you can see, this is a log graph on the you can see on the x-axis, it's a log scale. You can see that this concentration is very low. And so what's actually happening is that once you get the combination of APC and axon, you either recruit a GSK very quickly or you dissociate into APC and axon again. And so you essentially have here a trimolecular reaction in the sense that I was describing at the top. Okay, so here is sort of like a little bit clearer version of this small reaction here. So a axon, APC, APC axon, GSK, and the final destruction complex. And here's just a sample of what a simulation might look like if we did these bimolecular reactions using Smoluchowski approach. You would find that an axon and APC would eventually find each other, okay? And the moment they found each other, you would then say, well, GSK and, GSK and this complex react very quickly to create the complex, or it degrades. Now the problem is, at time t equals 0 when this is created, you then have a very fast reaction rate, which gives you a very large reaction radius, and regardless of the concentration of GSK, unless it's very dilute, you're going to get almost instantaneous recruitment of GSK, which goes against what we said in the first slide, where the Smoluchowski approach is not valid if you have very, very fast reactions. So in that very small time period where you're waiting for the distribution to give you a constant flux. Okay. So what I'm going to try and do is to look at how can we determine a rule for the sort of the relative positions of 3 molecules simultaneously, which give you a trimolecular reaction. So that's essentially what the problem is. Okay, so here is our example. I will look at an n-body Smoluchowski problem, but it's easier to talk about 3 molecules to start with. So I'll just remind you here that what we want is a condition in which these 3 molecules are sufficiently close to each other, but the condition that we place on that, whatever sufficiently close means, gives us back our reaction rate, K. So we have these 3 distances, and well, we don't really need 3 distances, okay? Because I could represent the relative positions of those 3 with just 2. So I'm going to look at the distance between 1 and 2, because of course if you had only 2 molecules, that's what Smoluchowski would have done, just looked at the distance between 1 and 2, and look at the distance of 3 from some point that has to do with this system. Now that could be the point x1, so I've sort of drawn it here. Actually, I've jumped ahead of myself a little bit. Before I do my analysis, actually, what I want to say is it's very easy to take a very naive approach and just say, well, we've got to come up with some condition. And like the two-body Smoluchowski problem, we just say, well, the most obvious thing to take would be the distance between two molecules, because we only have two molecules to consider. So now that we have these three distances, we could take these naive approaches. So we could say, well, what if we try to find this function sigma of k if our rule is that the distance between all three molecules is less than that distance, or perhaps the sum of the three distances has to be less than this sigma, or perhaps it's more of a like a Pythagoras sort of sum of the squares is less than the square of some distance. Who knows? Right? We're sort of just punting at this point. In actual fact, I think this first condition was tried out by Ocean and others, and they essentially they ran into a problem where they couldn't solve for sigma of k, and they had to take all these approximations, and then they could only really give a good result in like low dimensions and things like that, and only for three molecules. So the problem is analytic tractability, and this was one of the reasons why the Smoluchowski approach was so beloved, because we could actually solve for sigma as a function of k. Okay, so I'm going to come up with a more general rule. I'm going to say that we're going to define some function that's called, I'm going to call the proximity, so I'm calling it p. The subscript is just saying it's the proximity as a function of the three molecule problem. And I'm going to say that Pn is monotonically increasing for each variable. And this proximity of the three molecules, once this proximity becomes less than this distance sigma, which has to be a function of k, and I've got to work out this function, then I'm going to take my reaction. So, as I said, this Pn for the two-body problem is done because of we essentially set up a system which is analytically tractable. And so if we look at the Ford-Kolmogorov equation for this three-particle system, what we have is, so these x's are the coordinates of these three molecules, and we essentially come up with a sum of three Laplacians, if you like, which is not very nice considering that our boundary condition here, Pn, has to be a function of the relative distances between them. So first of all, we've got to do some sort of transformation, just like we did with Smoluchowski, where we transformed into the frame of A in order to find the relative distance. Okay. So this is just me rewriting down the problem, and I'm going to change these three distances to be vectors which are a little bit more nice, because I don't really need to describe them with three vectors. I can just do it with two. I can say eta1 is the position of the entire system. It's some sort of average position, if you like. Eta2 is the traditional two-molecule distance that Smoluchowski originally had in mind for two molecules. And this third one just says, well, let's take some position which is representative of these two and find the distance to the third one. This point here could be anything at this point. It could be this point here, it could be this point here, it could be anywhere along those that line, but it must be representable by the coordinates of x1, x2. So you end up with a Ford-Kolmogorov equation that looks like this. The hats here now mean with respect to eta instead of x. And this is not very nice. We want to find a analytically tractable answer here. We're not going to be able to have much luck unless we get rid of this mixing term here. This one here just says that the position of eta3 has to be, you want that to be independent of eta2 and eta1. Okay, all these vectors, you want them to diffuse, if you like, independently from each other. So we can choose this position here, which is how we're representing the average position of these two, and this position, which is the average of all three, in order to make this term disappear. Okay, and we come up with the center of diffusion. So the center of diffusion is quite a nice thing. The term, I didn't coin the term, I think Koichi Takahashi from who came up with EGFRD came up with that term. And essentially what it is it's like the center of mass, but instead of mass, you have the inverse of the diffusion constants of each of the molecules. And if you define these positions here as the center of diffusion, then this term here just drops out, okay? And you can show that just by doing the transformation directly. So that's nice. We're going to define these points with the center of diffusion, okay? That sort of helps us define what these ethers are. So now we just have anisotropic linear diffusion. And, so if our system is initially well mixed, it's not going to depend on eta. So we can drop out eta because it's initially well mixed. Okay, it doesn't matter what this vector is, it doesn't impact on the reaction. Okay, it's not in these boundary conditions at all. And then we can non-dimensionalize, and essentially what we do by non-dimensionalizing it is to make the anisotropic diffusion into isotropic diffusion in the new scaled space. So each of the dimensions will have diffusion the same in all of them. Okay, so we non-dimensionalize, and then the final step is to say we want to set up this Pn so that we can come up with something analytically tractable. So what we're going to do here is we're going to exploit symmetry. Here we have a 6-dimensional vector that has essentially a Laplacian in 6 dimensions. And we're going to, so we can set this up so that it's just a radial distance, just like Smoluchowski. So we're going to set Pn to be some factor, I'll explain what this is later, times the distance in the 6-dimensional space, and this 6-dimensional Laplacian just becomes in radial coordinates. So it's lovely. And now we can solve the problem exactly the way Smoluchowski did. So we can define, so here's our P3. Our P3 has a function of R. Remember, R was scaled, so all these ethers are scaled by D. So this is what the definition of the proximity is. And if we remember the condition for reaction is when the proximity of these three molecules is within this distance sigma. We can calculate the sigma by equating the reaction rate to the flux over the boundary over this 6-dimensional sphere. And so we end up with this thing. I haven't quite defined yet what this delta is, but I'll define that soon. Okay, so just a note about generalizing the problem. So we had a 3-molecule system. We're going to now have a 5-molecule system. So here we have our eta 2, our eta 3. Hello. Okay, sorry, I just went blank for a second. So eta 3 can be generated by taking the center of diffusion from 1 and 2 and joining a vector to 3. Then we find the center of diffusion of the whole lot, so starting from here and going out to 4. And then we find the center of diffusion again, which is here, and going out to 5. And we can obviously then, after we set up all our eta vectors, we can ignore eta 1. And we have a sort of a set of distances. Okay, so we set up our reaction rule to be when the proximity of these 5 molecules is less than sigma of K, and we can, this is just as easy, we can get the Kolmogorov equation, and the Kolmogorov equation, it will just be in 3 times n minus 1 dimensions as a sphere, and we can solve that quite easily. We can get our reaction rate in terms of alpha here, but alpha is just this function of n, which is the number of molecules, and this d hat is the relative diffusion of so di, for example, if I was 3, would be the relative diffusion of the molecule 3 from the center of diffusion of all of the previous molecules. And here is the definition of the scale parameter, and here is the definition of the proximity. Just defined the exact same way, just this sum of the squares of the distances that are scaled. So here we have our set. However, what's important here is we've defined the proximity of these 5 molecules as Pn, but they depend on these vectors, which we've sort of set up arbitrarily because we've labeled rather arbitrarily our molecules. So what happens if we were to relabel the molecules? Well, then all of our etas would then change to look like this. And one might think that this proximity would then change because we would fundamentally have changed our eters. But what's important is that, remember, we really want our proximity to be a function of the relative distances between the molecules, okay? And you can do, I'm going to skip quite a lot of geometry and algebra, and I'll jump straight to the answer. You can actually prove that this definition of proximity, no matter how you label the molecules, comes out to be this function here. And what this is if you understood the analogy between the center of mass and the center of diffusion, this would be the diffusion version of the radius of gyration. And so that's actually what the Smoluchowski radius is, or the definition of proximity is when you have more than two molecules. Okay.
So, sorry, I got
Yes.
lost. How did you reduce the system to just one spatial variable? What happened with all the? I understand you're at this Pn. I guess is your independent variable, and it's some combination of the etas. But why do all the angular degrees of freedom go away?
So could someone who's closer to the microphone repeat just maybe spot the
Never mind.
question?
Just continue. It's fine. Let's not derail you.
Okay. Sorry about that. The sound quality is not great in the room, I think. Okay, so this is just a summary of the result. The result is powerful essentially because the same equation that you use to analyze the Smoluchowski result with two molecules is basically exactly the same with larger number of molecules, just this power of R in the equation has changed. But essentially the boundary conditions are the same and it's more or less the same kind of analysis. So you can do all this analysis with pretty much everything the Smoluchowski theory has been applied to. So here we've got Doi kinetics. So instead of having a reaction on the boundary, you have it within the boundary with some particular rate. I've solved that problem, and it'll be in the paper that I referred to at the end. You can also look at what happens if you have finite time steps. So you don't have a really good Brownian simulation because you've got some finite time steps and molecules can jump in and out and miss reactions. You can do all the exact same analysis of all these papers and apply them to higher order reactions. So here's just a couple of numerical tests I want to finish up with. So here is just a test to see whether my simulation would work. So I set up a box that had periodic boundary conditions, so it's all closed, and I have the reaction A plus B plus C goes to A plus B. Essentially, I'm just using the trimolecular reaction to delete C, and I'm creating C at a constant rate of 10. Okay? So what we expect is that the distribution at any moment in time after it's reached some steady state for C, the number of molecules of C, should be Poisson distributed with an average of 10. Okay? So here is how the simulation would run. It might take a little bit of time to catch up. So when the molecule, the orange and pink molecule, which are the A and B molecule, get close enough together, within the realms at which a third molecule could be close enough to make the proximity of all three less than the reaction radius. And so eventually you'll see this green sphere pop up, and that's just essentially saying what distance does a particular molecule of blue or C need to be to the center of diffusion of the other two for a reaction to occur. And so the green sphere doesn't always even exist, which is really quite important. And I've run this simulation quite a few times, and I plotted the distribution of the number of molecules of C, and this is what I get. So I get these blue bars here. The Poisson distribution is these red dots. And what I find is that near the average, I get less probability of there being that number of molecules, and I get the tails actually get larger. And so what's going on here is if I'm already in a situation where I have a large number of C, I probably got there because A and B happen to be far apart. And it's going to take time for A and B, because they're diffusing in space, there's spatial temporal correlations, for them to come close together and be within a distance that will create this sort of green sphere. And the same with small number of C. This just so happens to be correlated to when these two molecules are close together, and you're going to have an increased rate at the rate at which the molecule is going to react. And I think this is actually not just a numerical byproduct, but something that which should happen with a real well-mixed system. Okay, so coming back to the Turing patterns, so here's our chemical system, and here is the spatial Gillespie solution. So here is the most common solution if you start off with the initial number of U as being the unstable uniform steady state. Here's the PDE solution, the dotted red line, and the histograms are the solution to a particular iteration of spatial Gillespie simulation. And they match fairly well in the sense that they're qualitatively the same. There's 2.5 peaks, essentially. And here's what you get with a Smoluchowski simulation, something quite different. In actual fact, whilst the spatial Gillespie simulation, you usually get these 2.5 peaks. With the Stochastic simulation, you get 3 peaks, and you don't just usually get them, it's quite stable, so you actually always get them. Which is a bit of a funny thing. So these bars here is the Smoluchowski simulation. And this is because when you come close to a boundary, and you have, for example, 2 U's close to each other, the V can approach a center of diffusion of these 2 molecules only from one side. They can't approach from the other side of the boundary because we've got reflective boundaries in this particular problem. This might be different if you had periodic boundaries, but this is definitely the case if you have a reflective boundary. In actual fact, that Steve Andrews, who is the developer of Smolden, has noticed this effect also with 2 molecule reactions, but they don't cause any macroscopic problems in a system because you need this trimolecular reaction in order to get the Turing pattern. So because you usually end up with something rather uniform near the boundary, it doesn't actually create that much of an effect on the entire system. But in this case, it does significantly change the entire Turing pattern. And you might be wondering about this red dotted line, which is the solution to the PDE, which is also stable. However, what I needed to do to create this red dotted line was to somewhat simulate these boundary conditions. So I needed to take K4, which is this reaction rate for the trimolecular reaction, and decrease it even ever so slightly. Here are the 2 parameters which I decrease it by on the 2 boundaries. And so I have these very small boundary layers which are created for the reaction rate, and the PDE then starts to agree with these Smoluchowski simulations. So it's an interesting question whether this actually would be what you would observe in a real Turing situation. So just some future work on this wind signaling. The reason why I don't have results to show you is, first of all, so here is remember the trimolecular reactions Axon, APC, and GSK to give you the destruction complex. So here we have the eta 2 and eta 3. So eta 2 would be the relative distance between Axon and APC, and GSK would be the distance between GSK and the center of diffusion of Axon and APC would be eta 3. And however, this is a reversible reaction, so you can go from this destruction complex back into its constituent parts. And so it would need to have the equivalent theory for reversible reactions. And I sort of know where I'm going with this, I just haven't done it yet. There's a paper by actually Kostas and Radek, who, I don't know if Kostas is there, I can't see, but some people that you may recognize have looked at this problem for the Smoluchowski bimolecular reaction. And so it just needs to be generalized, and I can do that. The other problem is with this sequestration, which doesn't happen. You can't have the interaction of parts of the molecule if you've only got 2 of them. Okay, so here we've got what happens if Axon and GSK combine to create something inert before Axon and APC combine? Okay, so what we essentially have is a new absorbing boundary that would create this inert complex, and this would take away from the flux that can come over the reaction boundary for the trimolecular reaction. So how do these sort of surfaces interact with each other is another question which I'm looking at the moment. So if you're interested in any of this stuff, I have a paper with SciAm, General Applied Maths. Hopefully it'll be accepted any day now. It's gone through a couple of iterations, but it is on archive, so if you're interested to have a look, that's where it is. So thanks very much for coming, and I'll take any questions. So was there a question at the back before that I could answer now, perhaps?
Don't worry about it. It's a stupid question. I figured it out.
So from your numerical simulations with three guys and the comparison between the stochastic simulations algorithms and the Smoluchowski one, you had discrepancies for the large tail and the beginning of the distribution, right?
So could you just repeat that? I caught most of it, but I just didn't quite get it.
So you had discrepancies between the simulations for the tail of the distributions, right?
Yeah, so you're talking about these or these ones?
The previous one, yeah, this one, yeah.
This one, yep.
So what if you increase the diffusion rates? Does the discrepancy vanish somehow, going to a more well-mixed model? Because so you claim that it's due to spatio-temporal correlations, right? So they should.
Okay, so the discrepancy here comes in because the rate of reaction is not. There's some spatial temporal correlations which aren't necessarily picked up with the law of mass action, which just says you just have to look at the number and you'll get the rate at which in the next small amount of time you'll have a reaction occurring. It also depends on the state of how A and B are initially set up. And if A and B are close, then you'll find that there's a correlation between when they're close and more molecules of C having just been reacted. So there's a correlation between a molecule of C reacting now and then shortly after. If that makes sense. I don't know if I answered your question. I hope I did.
I'll try. So what's the biological situation here? Are there really trimolecular reactions, or is it really sort of a transient complex between bimolecular reactions?
Sorry, I didn't catch much of that.
So in real biology,
You might have to talk into the computer. I don't know.
real biology, is it really trimolecular reactions, or is it really like transient complexes forming between two molecules at a time?
Yeah, so the whole idea, I mean, I hope I've got your question. So this is what I was sort of getting at here. The whole idea of sort of transient, this ABC axon, they're very short-lived, which means that this GSK, or the third reaction, is very fast. And so that when I'm talking about a trimolecular reaction, I'm really talking about these sort of catalytic reactions where one sort of occurs, and as a result of that occurrence, you have a very fast reaction as a result. And it's this very fast reaction that causes the Smoluchowski model to break down. Because you essentially say, well, it's not fast, it's more or less instantaneous. Because this reaction radius is so large, it can pick up this GSK, this GSK, this GSK, or this GSK. It doesn't really matter where it is, because you're essentially looking very far in space to find it. And because it's virtually instantaneous finding it, you're in a situation where, if I go back to the start, you're in a situation where you're at time t equals 0 when you create your reaction, you're absorbing reaction radius, and the rate of reaction is essentially very, very large. So it's not the rate, it's not this fast reaction rate which you hope to simulate. It's almost infinitely fast. So the reverse reaction virtually never happens. So this fast reverse reaction never happens. So essentially you're limited by the concentration of axon and ABC going very slowly, and then an instantaneous reaction. So what would be the end result of this? Well, essentially you would just have, you wouldn't have law of mass action for a start, you would just have a reaction rate that depended on the concentration of axon and APC, and then that instantaneously created this 3 molecular complex.
Okay.
So that's the problem, at least from a practical point of view, with that interpretation. Of course, from a physical point of view, this is exactly what happens. It's just from a simulation point of view, or it's not very useful for us.
Okay.
So Mark, what makes the second reaction fast physically? Like biologically, why is that reaction so fast? Why is it faster than diffusion limited? And I'm confused about
Yeah,
that.
okay, so for this particular example, I'm not a biochemist, so I'm not sure, but for most of these kind of catalytic reactions, you typically find that the molecular structure somehow changes to allow for GSK to come in quite fast. Yeah. So some sort of like molecular structure thing, which unfortunately is a bit over my head because I'm not a biochemist.
But it's coming in faster than the diffusion limit?
And also someone has to be there.
I mean, if they're all over throughout this, like there's some encounter limit,
It
right?
starts in the well mixed state.
Even if you start well mixed, there's some encounter limit based on how much GS.
Well, it is a protein-protein interaction, and typically these are, I mean, there's a doubt that there's some sort of long-range attraction or anything like that. It may be because GSK is in large quantities, but it's in no more larger quantity than, say, APC and axon. If you look at sort of the relative amounts here, you've got axon, that one's a bad example. No, actually, no, it's not too bad. So you've got axon here in blue, and APC is relatively the same amount. That's about 5 times more, well, actually, probably 10 times more, actually. But it's not significant orders of magnitude difference. So it could be if it's reaction limited, but the concentrations aren't that great. And then our proteins are diffusing around about the same amount. APC and axon is slightly larger, so they would be diffusing slower. But I'm not sure actually. Yeah, sorry, I can't answer that one.
Okay.
If there are not any other questions, let's just thank Mark again. And let him go to sleep.
Yeah. Good night.
Good night,
Good
Mark.
morning, maybe.
Have a good afternoon, everyone.
Yeah, thank you.
Okay.
Bye-bye.
It's 2 a.m.